Welcome to another episode of Mechanism of Action. I'm your host, Adam J. Brown. Today is our last in the series of asthma. So this is we've done multiple episodes involving asthma and different mechanisms of actions of multiple medications and the story behind them. Today we're finishing off that series before moving on to new disease in the next episode. So I hope you've enjoyed the series on asthma. This episode I've been looking forward to putting together for a while after I stumbled upon the story, and the story involved a horse. And what was very intriguing to me is how this kind of story evolved over the course of over half a century. I think many of you have a vague understanding of IgE. It's an antibody that springs to mind whenever you hear about an allergic reaction to something. So I think you're going to appreciate the story of the story of allergies a whole lot better after hearing this story. And you're going to be able to comprehend what's going on from multiple different allergic standpoints after the end of this episode. So this episode is called IgE, Horses, Fish Scale, and the story of elucidating the mechanism of allergies. We start in the year 1919, summer, in New York City. We present a 35-year-old man from Greece, now in New York, who worked as a waiter beforehand. He was feeling run down and diagnosed with anemia just a few weeks before. The diagnosis isn't clear what caused anemia is. All we know is he had anemia. And before this diagnosis, he was healthy. No history of rashes, allergies, asthma, nothing. He was around horses quite a bit. He used to use them to navigate around New York, for example. So back to his poorly defined anemia. He was feeling worn down. He was given a transfusion of blood, which helped him pep up some. So back then, they just gave him some blood from a donor, and his hemoglobin rose, and he actually started feeling better. 2 weeks after the infusion, the man went on his normal routine, and he went to Central Park. And he decided to get into a horse-drawn carriage to get around. Shortly after stepping into the carriage, though, he noticed a sensation, something different. He felt his chest tightening. He paused. He sat in the chair. His eyes shifted left and right, anticipating how this tightness may progress. He had never felt it before, and then he noticed the trouble drawing air in. He struggled. He couldn't get air, and he had to pull it harder and harder. His heart started to pound. He could feel his heart rate racing through his chest. The tightness increased, and he asked the carriage rider to take him home immediately, where he walked in and laid on the floor and had to have his doctor called in. And they did home calls back then. Over time, his breathing and chest slowly improved once he was home, but by the time the doctor got there, he had considerable wheezing in his lung field, and the doctor opened up his black satchel and injected him with subcutaneous epinephrine. Within seconds, he could feel this tightness across his chest alleviate and his breathing improved. He felt well, but understandably very confused. After the doctor told him he suffered an attack of asthma, he had never had asthma before, and he was in his mid-30s. Isn't that something younger people get? What's going on? But after a few days, well, he was back to normal. He wanted to get on with his daily routine. He went back to Central Park. He walks close to a horse, and once again, boom, suddenly he is seized by tightness in his chest and difficulty breathing. As quickly as he can, he leaves the park, and he slowly feels better. He goes to his doctor, who thought he might, again, have an allergic reaction, and he actually does a skin prick test check of various things like pollen, cat hair, all negative, except for horse dander. So a skin prick is where you have like a small little protein at the end of a pin, like a needle, and it could be a cat hair, dander, pollen, and then you prick the skin. And then you see if that area kind of lights up. It's called a wheel and a flare, where it kind of raises and gets red. And erythematous, it's kind of allergic reaction, allergic response. So he said, oh my goodness, why is this? I'm in my mid-30s. Why all of a sudden am I allergic to horses? This is crazy. And the doctor said, this is very odd. And he realized, wait a minute, didn't you have a blood transfusion 2 weeks before? And the patient said, yes. So the doctor actually reached out to the person who donated the blood. Back then it was a 1-to-1 transfusion. It wasn't just a mixture of blood. And the patient came in and said, oh yeah, I have multiple allergies. So the doctor actually did a skin prick on him, and lo and behold, he had a severe allergic reaction to horse dander as well. This was the first realization that something from an allergic standpoint can be transfused to somebody else. So the doctor is shocked. He never heard of anything like this. He knows infectious organisms can be transfused, but an allergic reaction, this is bizarre. Allergies, by the way, was just starting to be elucidated in the century before, so it's very much not fully understood. Allergies were not nearly as common as they are today, so it's very unclear understanding of what was going on that was actually even causing allergies, let alone something that can be transferred via blood transfusion. The doctor said this must be some sort of anaphylactic bodies that are transferable. He wrote a paper in JAMA cautioning doctors from using blood of patients with severe allergies and blood transfusion because of this. At this point, we do not understand the mechanism of what could be causing this, what could actually be being transfused within that blood that can actually transfer the allergies this patient suffered from. Then, just a few years later, because of this paper that came out in 1921, two German physicians, Dr. Heinz Kussner and Dr. Karl Prosnitz, were interested in this phenomenon of transferring allergies that was reported just a few years before. So Kussner apparently was very allergic to fish, or Fisch in German. He decided to take advantage of this allergy and demonstrated transferability in a controlled fashion. The Kussner drew up some blood of his own blood, spun it down so he could exclude the red blood cells to prevent an allergic reaction. Then he drew the plasma and injected it subcutaneously into that of his partner, Dr. Karl Prosnitz, who probably wondered if he tasted his last yak, which is German for salmon. Ha! See, he was wondering if maybe he also is going to be allergic to salmon. But again, it wasn't a blood transfusion, it's a subcutaneous injection to see if he might have a response in his skin. So Kussner waited 24 hours, then got fish. Unclear what type of fish was used. And he ground up the scales and then actually put it on the tip of a needle and then inserted it into the area that was injected with the plasma, and then the control arm where there was no plasma injected. And lo and behold, very quickly there was a will and flare response in the area of the skin of his partner. This was showing that he had some transferability within the plasma of his fish allergy. So this was a big deal because it actually allowed people to start using this reaction, this type of fish transfer reaction to the skin, to start being able to measure allergies in a controlled fashion and start doing experiments with it. But the big question at this point was, what is being transferred? So some early theories are maybe it is a type of antibody. Well, you can confirm if it's an antibody by using temperature, basically. This is because different proteins will denature or unfold at different temperatures. You can take the plasma of a person who's allergic to fish, for example, and then heat it up to various degrees and then see when it stops transferring. So all of a sudden, when you inject it to the skin of somebody else and it no longer reacts to the fish scale, that's telling you that whatever temperature you brought it to, it denatured that protein. And when they heated up to the temperature of denaturing antibodies, which was at almost the same temperature of IgG, the fish transfer stopped working. So they had this hypothesis is probably some sort of antibody that was being transferred, that actually transferred the allergies between these two people. So we got a clue that this was an antibody based on the temperature, based on the acidity and salt concentrations that these experiments were looking at, and they had similarity between IgG. But IgG, for example, we knew didn't actually cause hives and cause a wheel and flare like we saw with whatever was being transferred with the fish. So this next antibody wasn't identified until 1960s. And the reason was because it's actually extremely scarce compared to IgM, IgG, and IgA. IgE makes up about 0.005% of all immunoglobulins. So this made catching it with analytical methods in the 1930s, 1950s almost impossible. Another fun fact about IgG is it's quite transient. Its half-life is only 2 days in the blood if it's not bound, versus IgG, which is 21 days. And it turns out, we'll skip ahead a little bit, when IgE binds to a mast cell, which I'll get to in a minute, then it can last there for weeks to months. It can be bound to the mast cell, but that would not be measurable in ways people were doing in the 1930s, 1940s. You couldn't measure it when it was bound to the mast cell. So they're measuring mostly the free-floating form, which is very minute quantities and also very short-lived. So it's very, very hard to catch what IgG was doing. Then we get to 1967, when we get doctors Kimishige Ishizaka and Dr. Teruko Ishizaka, who were working at the Children's Asthma Research Institute and Hospital, which was later named National Jewish Hospital. They designed a series of experiments, DEAE cellulose chromatography and gel filtration. They used to separate proteins, isolated dozens of fractions of proteins, and then took the fractions of proteins and did the fish test again. Next step, they were able to remove IgG from the fractions, and the fish test still worked with no IgG in whatever the plasma was being transferred. So they really were able to find, hey, this is not IgG that is doing it. These doctors with this newer technologies were able to take away all the IgG, and whatever we're giving is not IgG, and it's still positive. So finally, they were able to isolate the protein and realized it was a similar size to IgG, but then it behaved very differently. At the same time, these two doctors elucidated what we now call IgE. Physicians in Sweden were also stumbling upon it. They were stumbling upon it in a very different way because they were trying to understand a patient who was presenting with myeloma. The myeloma is a relatively common type of blood cancer that causes an uncontrolled, massive malignant production of immunoglobulins, primarily IgM or IgG. But in this patient, they had this massive amount of antibodies, but it wasn't IgM, IgA, IgG. Like, what is it? And long story short, they're actually able to realize there's a new antibody. And they got together with the Japanese colleagues from National Jewish Hospital, and they called it IgE. Now we have IgE, and they're able to measure it. They're able to elucidate it. But the problem is, as I mentioned before, it's still in very minute quantities. It's very, very difficult to actually do experiments with it because it has such a small amount. So how are they going to do more improved experiments with it? Well, they actually had another patient who developed IgE myeloma. It's an extremely rare type of cancer. But this patient recognized he had it. His name was Peter Shackford, and he actually donated 30 liters of his own plasma, along with his treatments with plasmapheresis. He did this for 1 year before he died to help science better understand this strange antibody that he was producing in a malignant level. So thanks to his sacrifice, we actually understand IgE a whole lot better because of all the different experiments that can be done, because all of a sudden now we have an abundant source of IgE, and we're able to rapidly accelerate the understanding of this antibody. So the first step was to prove, hey, is IgE what we were looking for? They were able to detect it. They realized it could be associated with different type of myeloma, but is it really the transferable agent? So they got their answer when they were able to isolate IgE in a patient who had an allergy to ragweed, and then take the plasma, take the IgE out of that patient with ragweed, and inject it subcutaneously into another person. And lo and behold, that person now had an isolated ragweed allergic reaction with a wheel and flare into their skin. So this is a big deal. Lightning crashes, fireworks go off, say, hey, we have this major component of allergy that we now can recognize, but we don't know the whole story. So now we have to turn to those other two major puzzle pieces, which is histamine and the humble mast cell. So then we'll get back to IgE in a minute, but take a step back to get these other puzzle pieces that have been known for a while, but didn't really come together until the IgE discovery. Histamine was elucidated well back into the 1910s. We didn't know where it came from, but we were able to isolate it and inject it and recognize that when it was injected to different animals, it would cause vasodilation, hypotension, uterine contractions, and when you injected it into skin, it would cause a wheel and flare, that raised skin, the erythematous, itchiness associated with it. So there's a strong association with histamine and allergies way back then. They realized that a lot of the signs and symptoms when histamine was injected intravenously literally mimicked anaphylaxis. Patients would also have bronchospasm and chest tightness and difficulty breathing. So they realized that histamine clearly is this major driver of allergies. But they didn't know where it was stored. That knowledge came when they discovered the mast cell. But the mast cell was discovered actually in the late 1800s, so well before histamine was out. And it was discovered by none other than Mr. Paul Ehrlich, who would later win a Nobel Prize for his massive contribution to immunology. But I didn't say Dr. Paul Ehrlich because when he discovered mast cells, he was actually a medical student. So you're just like in the lab working with brand new dyes. So it's like just like most medical students doing research in a lab and developed a groundbreaking study and wrote it up in a paper before they became a doctor. It's pretty typical. So he made this groundbreaking discovery and wrote it up in paper well before he got his MD. So he's a big deal, but one of his initial discoveries I didn't know about was mast cells, because he was very big from a pathology standpoint. He was really interested in dyes and how different dyes can highlight different cells. And he was intrigued by mast cells. I think what caught his attention is these cells kind of attached to various tissues, and they had lots of granules, these kind of little balls inside of them. Because of these granules, he supposed the cells had lots of nutrients. I think he thought that these granules were probably lots of nutrients stored, and they were kind of helping feed the rest of the tissue, which is incorrect. So he called them Mastzellen, which is German for apparently to fatten or feed, because he thought the cells looked fat. Fat shaming in the late 1800s. We don't tolerate this anymore in our pathology practices. No more fat shaming cells, okay? But long story, the granules were not fat or not nutrients. Turned out to be histamine. Spoiler alert. It wasn't until 1953 when we got some indirect evidence that it stores histamine coming out of St. Andrew's Medical School in Dundee, Scotland. where tissue was collected from various animals and mast cells were quantitated in each tissue, and when the tissues were subjected to histamine liberators, which are chemicals who kind of do as described the trigger histamine release. Okay, so they're looking at these animal tissues and it said, hey, look, I want to see which tissues liberate the most histamine when these histamine liberators were injected, okay? And lo and behold, the tissues with very few mast cells in them, when they injected the histamine liberator, hardly anything came out. So hardly any histamine came out. In contrast, tissues that had large quantities of mast cells, abundance of histamine was released. So this is kind of indirect evidence that, hey, maybe these mast cells are actually what's holding the histamine and allowing the histamine release. So another study in the same paper looked at what if we take a tissue that has lots of mast cells, such as the liver of this animal, and incubate it with these histamine liberators. And they, shake it about with the histamine liberator and look under the microscope, and those mast cells that is full of actually all no longer had the granules inside of them. And there's a lot of histamine liberated and floating around in the fluid around it, but the granules in the mast cells had disappeared. So they kind of said, oh, that's probably where the histamine is coming from. That's actually part of the granules that we see in mast cells. It wasn't fat. Poor mast cell. Don't feel shame anymore, okay? It was histamine. So we have, 1953, we have an understanding of histamine, recognizing it's associated with allergic reaction. Now we understand where histamine is coming from mast cells, but we don't know how mast cells get triggered to release histamine. But in 1967, we get IgE as the transferable agent of allergies. So maybe IgE has something to do with the mast cells that trigger the release of histamine. So that's kind of how that came about, okay? So injecting IgE into the skin does not cause an allergic reaction by itself. So an allergen has to be added, and that's when the will and flare develops. So we think this allergen triggers the IgE, which somehow has something to do with the mast cell, and the mast cell triggers the release of histamine. First step is to see, do IgE and mast cells interact? So in the decades of the 70s and 80s, we have a technology to do this, which is called radiolabeling, where you can radiolabel IgE and then send it off into tissue and then be able to detect where it lands in that tissue with the radioactivity. Lo and behold, the radioactive labeled IgE were abundantly attached to the mast cells. And the researchers did the experiments with IgG, IgA, IgM, and like me at prom, none of these antibodies bound to the mast cell. So the mast cell was unbounded, was all alone. So the mast cells wanted nothing to do with IgG, IgA, IgM, such common folk, apparently. So eventually the researchers discovered the unique receptors on mast cells, and they were able to elucidate some very important components of how the IgE interacts with mast cells. They found out, A, they bind extremely tightly. Once IgE was bound to it, it was there for weeks, or potentially to the life of the mast cell. There's tight binding and extremely slow dissociation. So that helped explain why if the patient had a transferable allergy, such as that fish, they could check like a week later, 2 weeks later, that patient would still have that allergic reaction to the same spot. It also explained why IgE was so hard to find in circulation, because hardly any of it is free floating in the fluid. Almost all of it is bound up to the mast cell. But we still haven't explained the other component of this puzzle, which is the allergen, like fish, like ragweed. How does this interact with IgE and the mast cell and histamine release? So another study came about looking at measuring histamine fluorometrically, and they actually measured histamine release across 4 states. Mast cells that had either IgE, had no IgE, had IgE that was sensitized to ovalbumin, which is kind of a trigger, or IgE not sensitized to ovalbumin. And they looked at all these different combinations where they just give it IgE and the mast cell, and then lo and behold, without an allergen to activate it, IgE bound to the mast cell, but no histamine was released. Then you'd have IgE that was exposed to and sensitized to ragweed, and that would bind to the mast cell, and then they would expose it to ovalbumin, and nothing happened. Then you'd have a IgE that was sensitized to ovalbumin, which is this kind of large protein from eggs, and then that IgE was bound to a mast cell, and then you expose that IgE to ovalbumin, and then lo and behold, that triggered. So the combination where they had the ovalbumin sensitized IgE, once bound to mast cell, and then you expose it to ovalbumin, that would trigger that sequence event, the ovalbumin binding to the IgE, IgE triggering the mast cell to degranulate and release histamine. So that was the aha moment, right? So we have, okay, this IgE has to be specific to the allergen. And we also found out that mast cells actually could have tens to hundreds of thousands of receptors on a single cell, which is kind of literally hard to visualize. And as a result of this, one mast cell can be bound to thousands of different types of IgE. Another interesting fact about mast cells and IgE is they found that floating IgE regulates the amount of receptors present on mast cells. So if a lot of IgE is floating around, the mast cells will upregulate their receptors to accommodate more IgE. But if there's less IgE floating around, the mast cells actually downregulate their amount of binding sites because there's not as much IgE floating around. So theoretically, if you're able to massively reduce the amount of IgE floating around in a patient who is allergic, you could potentially really, decrease the triggering of IgE binding, but you also decrease the amount of receptors available to the mast cell. So you can really decrease the allergic response from twofold. The researchers did something clever. They realized the Fc portion of IgE that binds to mast cells has a different protein conformation when it's not bound. So what they did was they gave this medication that actually binds to the IgE that was still free floating. And if you kept giving it, you kind of bind up all that IgE before it binds to the mast cell. So that was the idea. So the drug was named omalizumab and is used for the treatment of asthma. So a key clinical trial investigating this was a phase 3 study looking at it. It was called the treatment of allergic asthma with monoclonal anti-IgE antibody. 317 patients, wide age range of 11 to 50, with moderate to severe asthma. 3 treatment arms: high dose anti-IgE, low dose anti-IgE versus placebo. And the drug is now subcutaneous, but at that time the trial was IV. And they found that the serum-free IgE fell by 95%, so a massive reduction in the amount of free IgE. And the patients on the drug in the active treatment group, they required less inhaled corticosteroids, they had less hospitalizations, they had better improvement in breathing. So it was pretty clear this drug had a major impact, and it demonstrated how important IgE is in the treatment of asthma and the pathophysiology of asthma. So it actually opened the door for utilizing anti-IgE in a variety of medications. Next was 2014, chronic spontaneous urticaria; 2020, chronic rhinosinusitis with nasal polyps; 2024, IgE-mediated food allergy. So multiple diseases are now being treated with this medication, and I've seen a lot of patients on it with a significant improvement in quality of life because how much less rashes, less urticaria, less wheezing, less need for inhalers because of better control of their asthma. So it can make a huge difference. One last thought before we stop this, something that doesn't really cross my mind much until I was writing this, and I'd never thought of it before. And I asked myself, evolutionarily, why do we have these cells, mast cells and IgE, right? So, what evolutionarily good did anaphylaxis do, or having hives associated with cat? What benefit did that give? And we don't really know the answer. We don't know the answer to this, but we have some ideas, mostly through recognizing that, like, when there's knockout models of mast cells, animals tend to have higher risk of parasitic infections. So there was probably some evolutionary purpose back in the day. Mast cells are probably pretty important for different infectious organisms, including parasites. They're abundant in the intestines, so it may have helped clear parasites. Animals without mast cells have higher likelihood of dying of these parasites in these controlled experiments. Also, higher risk of dying from toxins like venoms from snakes, for example. So the mast cells may have had something to do with those responses to parasites and venoms, for example. Mast cells have been around much longer than IgE, so that their interaction was a much later step in evolution. For example, multiple types of non-mammalian species in nature, such as fish, amphibians, and birds, have mast cells, but they do not have IgE. So mast cells are much more evolutionarily ancient compared to IgE. So this interaction occurring between the mast cell and IgE is probably a much more recent interaction in the grand scale of human history. And it was probably just an accidental bystander response and not the primary reason why they exist. But in whatever we're doing in our modern lives, this has become their predominant function. That's why people think about the hygiene hypothesis, that maybe we're too clean and we have all these cells that were developed to kill parasites all the time, and now we don't have parasites anymore, so now they're bored and they're causing allergies. So that's a whole other story. But I hope you enjoyed learning about IgE and mast cells and allergies, and I thought that horse story was really cool, and it really helped me understand what these antibodies are doing. So thanks for listening.